Good morning, everyone. Good afternoon and good evening, and thank you for everyone joining us today. Thank you as well for making time to attend EnviroZyme's technical webinar, Optimizing Refinery Wastewater Treatment, a Biological Approach. EnviroZyme is a global leader in fermentation and the development of biologically based solutions for wastewater applications, and we're excited to share this session with you. Our goal today is to provide practical technical experience on several key challenges commonly seen in refinery wastewater, many of these which will also occur in other industrial wastewater treatment systems. My name is Michael McWilliam, and I'm the Sales Director for EnviroZyme in Asia, and I'm based in Japan. Presenting the technical content today is Matt Livingston, EnviroZyme's Global Technical Director, joining us from Virginia, USA. Matt, thank you for being here, especially as it's late evening for you at your time. Before we begin, a couple of quick housekeeping items. The session is planned for 1 hour, and for confidentiality, attendee details are hidden and only Matt and I will be visible. Audio participation is disabled, so you won't be able to speak or raise a hand during the webinar, but please submit questions during the seminar through the Q&A tab at the top of the screen that you'll see. I'll collect these, and at the end we'll cover them off with Matt through the Q&A session. If helpful, you can also enable the speech-to-text translation. So that is the More box at the top of the screen at the right side, and you scroll down to Language and Speech option, and then you can choose your local language for the speech-to-text translation automatically. We'll also be running a few live polls during the session, so please feel free to participate in those when they appear up on your screen. With that, I'll hand over to Matt, and I'll say again, thank you for everyone joining today, and we look forward to supporting you and your customers with any refinery or industrial wastewater challenges in the future. Thank you.
All right, thank you, Mike, and thank you everybody for taking the time to join us this afternoon or this morning for you. We're going to be focusing today mostly on refining wastewater treatment, but a lot of this information can really be applied across different industrial types of applications. So if you're treating wastewater that's not refining, there's certainly some things I hope that you can take away from today. We lead with biology because our platform is around biology. It's around bioaugmentation for biological treatment. And refineries and a lot of industrial wastewater, but refineries in particular, can contain a variety of hydrocarbons, phenols, amines, sulfides, and other organics that cause issues that are suitable for biological solutions. So that's why we feel this is a good application space for bioaugmentation. And we'll talk about a couple of those key applications. Before we get into that, I'd like to do a brief introduction to our company and our bioaugmentation platform, just so that we're all starting in the same place. EnviroZyme really is the world leader in bioaugmentation. We are a subsidiary of the Betco Corporation that's headquartered in Bowling Green, Ohio in the US. We have a GMP ISO certified FDA registered facility where we ferment, blend, and package a variety of microbial solutions, both custom solutions and off the shelf in our product line. We have local sales and technical support covering North America, Latin America, Europe, and Asia Pacific. At the end of the presentation, I'll share the contact information for the different folks. And we work our solutions that get down to the end users through global and local partners. So some of our partners have a global scope, but we also work a lot with local partners in different places around the world. We're very excited. In Q1 2026, we are opening a production expansion. You can see our facility on the upper right-hand side, and you can see the newer roof on the left-hand side of the building, where we're essentially doubling the capacity of our fermentation. EnviroZyme is really the product of a couple different acquisitions. In 2009, Betco acquired Biosystems International and then rebranded that into EnviroZyme in 2023. They acquired the wastewater business from EnviroZyme. I've worked with EnviroZyme for over 20 years, so have a lot of experience in this area and come from that acquisition. And then just this past in 2024, acquired the wastewater business from BioWish. So rolling all these companies up make us the largest company in this area and have the largest fermentation capability in this space. What we do in the production side is we start with organisms that are naturally occurring, that are found in nature. We streak them out on a petri dish and take one colony, so we know that it's very pure. We put that into a growth media in the flask, to a larger seed fermenter, to a larger fermenter. And in our factory, this is a picture of our 30,000 liter fermenter. We'll grow it up in that fermenter one strain at a time so that it's pure. We'll centrifuge it down. In some cases, we'll spray dry that material so it's very concentrated. And we can even use that as a pure culture or as a raw material that we can then formulate into different types of either powdered products, solid products, or liquid products that we bring out to the market. When we talk about our bioaugmentation platform, that is the practice of enhancing the indigenous or native bacterial populations through the addition of a concentrated natural bacterial strain with specific degradation capabilities. And those different capabilities will be different depending on the application or the market. We'll use different products in food processing than we will in refining, and different products in pulp and paper, again, because the target organics are different in different applications. So that's a way we differentiate our products. We think about four main applications for bioaugmentation, the first being a startup, or when a plant is being commissioned or after a turnaround, trying to get that efficiency of the wastewater treatment plant up to its maximum level as quickly as possible. And then the system efficiency is another application where we're trying to improve the overall efficiency or COD degradation within the existing equipment at a plant. What we often see is that when a plant is new, it builds a wastewater treatment plant on it. Often the factory will increase in size and the wastewater treatment plant will remain the same size. So over the course of time, it often becomes undersized. So that's a good application for bioaugmentation. In a lot of industrial processes, there's a lot of variability causing stress on the wastewater treatment plant. A lot of times that stress becomes too much and the plant becomes upset. So we try to maintain stable treatment with bioaugmentation. And then if there is an upset, and upsets inevitably happen, there's an upset recovery application. And the goal there is similar to the startup. We're trying to get that performance back up to its maximum level as quickly as possible. So that's a short overview. I think a lot of folks maybe have some background in bioaugmentation, but that's just a brief introduction. I want to talk next about why refineries are good candidates for bioaugmentation. Crude oil, again, is the source of all refineries. That's the feedstock. And it's a very complex mixture of hydrocarbons and a lot of other components. And those components will vary by deposit and location. When I started in this industry almost 25 years ago, or over 25 years ago, there were not as many sour crudes on the market. It was mostly sweet crudes, at least the refineries that I was going to. But sour crudes now are cheaper and more abundant, and refinery technology now allows sour crudes and cheaper crudes to be processed at a more profitable rate. So sour crudes would be somewhere between 0.5% and 5% sulfur content. That would be the definition of a sour crude, or a heavier crude that has an API gravity below 20 degrees or less than 20 degrees. Sour crudes and heavier crudes were not that common 25 years ago. And when they started showing up at different refineries, there were a lot of problems, a lot of biological issues, a lot of stress on the plant. And there's been a lot of innovation allowing refineries to now handle this material in the way that they weren't able to do it, say, 15, 20 years ago. In a report that I found towards the end of last year, just looking at who's refining heavy and sour crude, really everybody globally, all the major players in North and South America, throughout Europe, and also in Asia. I guess this is more focused in Asia. So some of the refineries and companies listed in this paper were S-Oil, Reliance, Sinopec, and Fujian Refining. A lot of players across. These are just the ones that were listed in that paper, and I know it's a lot more than this, but it's very common now to see these. So with that, I do want to get into a poll, and if you are so inclined, it might take a second to load up here. So when we get into refinery waste streams, there's a variety of processes, and no two refineries are exactly the same, but there are a lot of these units, again, that are seen in a lot of places, and the key contaminants that come up are often toxic at high concentrations, but most of these are degradable and can be managed through biological treatment. So at a lot of refineries, that's why they have a biological treatment plant at the end of the collection system, and it's these streams that go in for treatment. Before we get into the biological treatment, it's really important to focus on effective pre-treatment. It's really the last defense before this material gets down into the biological treatment. So it's really worth optimizing and investing and maintaining and cleaning out the API separators and the DAFs and equalization. Those can be arranged differently at different refineries. But it really is important to try to maintain good pre-treatment. That's really going to help the biological unit. Again, the less variability, the less stress that comes into the plant from changes, the better off the biological system is going to be. When we get into biological treatment, to make it really simple, we think of two primary goals for wastewater treatment. One is for the bacteria to remove pollutants from the water. And then the second is to form a dense flock that settles and can be removed. So here you see a picture on the left of just bacterial cells. During the treatment process, you hope that they clump up and form a nice flock that you see in the middle. And then in the settleometer picture, you would love your clarifier to look like that, where you have a nice dense flock, settled biomass at the bottom, and a very clear effluent coming out of the top of your clarifier. But that is not always the case. And it's really hard to have that be the case all the time. There are a lot of growth pressures in a refinery and in wastewater treatment that impact the flock. A lot of these cause stress. So it could be inert material that could be coming down through the process that doesn't get removed through pre-treatment. There can be competition among different types of bacteria. There can be bacterial filaments that can outcompete some of the flock-forming bacteria. There can be toxins that come in that can be toxic and cause stress or kill off some of the bacteria. The sludge retention time, how old or young the sludge is. The sludge is going to behave, the biomass is going to behave differently. It will grow faster or slower depending on the sludge age. pH, especially in a refinery, can vary if that's not controlled. The types of substrates that are coming in, dissolved oxygen concentration, and temperatures, either high temperature or low temperature. But all those can cause stresses on the system. And then operations is really the key to managing those growth pressures to making the plant run as effectively as it can. So I'm going to try another poll. Hopefully this one will come up a little easier. This is about persistent biological issues at your plant. And let's see, back to the question. Hopefully this can. Okay, well, Mike, if you're on the side, if you can look into that, we're going to move past that one and see if that's okay. So the next we'll talk about is troubleshooting some of these issues that we often see in either industrial wastewater treatment or in refining. And the first is really understanding the data and visiting the site and understanding how the plant is operated. And plants will have all sorts of data from mixed liquor, suspended solids to settling data and BOD influent and effluent, nitrogen, phosphorus. It's really key to looking at all of that data and understand, first of all, is the right data being collected? And if there's a problem that the plant has, can you see where the problem is described in the data? Because if you really can't see from the data what's causing the problems, it's hard to come up with a solution. So trying to figure out, again, in the data, what's described in the problem, so you get a sense of, when you can make a change, you might be able to see the data also improve for that. So it's really getting on the site, understanding the layout of the plant, getting a good plant diagram, walking through, making sure the equipment works, making sure the plant physically is in good shape, and then understanding, again, from the data, what's causing some of the issues. The second is, using a microscope and doing a microscopic examination. And we could do a whole seminar on a microscopic examination. It's really a way to get a lot of information about the plant. It's looking at the flock and getting an understanding. Is it a good solid flock? Is it open? Is it small, dense, loose flock? That's the first. The second is filamentous bacteria. You can have filamentous bacteria growing from flock particles that make it hard to settle, that make it hard to compact. These filaments, we'll talk a little bit about them later, have different causative factors. And if you can identify the filament, that can help orient you to what the factors are that you can try to change to improve the situation. Looking at the higher life forms, there are small single-celled and multi-celled organisms other than bacteria that are in your plant. And understanding those higher life forms can also give you some information about stress and sludge age and other things about your system. And then looking at the bulk water, is outside of the flock particles, is the water clean? Is there a lot of suspended bacteria? Is there a lot of inert material? You can really get a lot of good information from doing a micro exam. The third level is something that's a little more newer that we're seeing more and more out in the field, is doing DNA analysis, doing community analysis on the organisms that are in your system. This technology has come a long way in the last, I would say, 5 years in terms of not only becoming less expensive and easier to do and quicker turnaround, it's getting that data and being able to make sense and using that data in an appropriate way. There are people that do this all over the globe. We have a partner in the US called Aster Bio that we work with. And typically what happens, we send them off a sample. They identify it down to the genus level. So in biology, you have the kingdom, phylum, class, all the way down to the genus and species. They typically go down to the genus. There's typically hundreds of genuses in a sample, and they separate it into functional groups. So whether it be ammonia oxidizing bacteria, nitrate oxidizing bacteria, sulfur oxidizing bacteria, filamentous bacteria, foam causing bacteria, or EPS causing bacteria, it can give you a good sense, in particular around some of the problems like filamentous or foam or EPS that we're going to talk about soon. So we're doing more and more of this, and if you have a chance to talk to somebody in your region that's doing this, I would encourage you to pursue that. We talked about some of these key bioaugmentation applications in refineries, or in general. This specifically is around refining. So again, for a startup, after a plant turnaround, that's when we're often looking at the startup type application. System efficiency can be around design issues or temperature. It can be around BOD or ammonia or TSS or phenol, hydrocarbons, trying to improve the degradation of these compounds. When it comes to stability, again, it's often the feed variability that's coming down from upstream in the refinery It could be changing the crude slates. That could be causing some stability issues or toxicity, causing settling issues, temperature stress. And then upset recovery is often a nitrification. We'll talk about nitrifying bacteria in a little bit. They're a little more sensitive than some of the other bacteria. And if you lose nitrification, you need to make some changes to recover that. Or recovery could be from toxicity or even hydraulic washout. So different issues can cause upsets at a refinery. Okay, now specifically in refining, we're going to talk about a couple issues: EPS, nitrification, and then some comments on sludge settling. So EPS is extracellular polymeric substances, and that's becoming a real big issue, especially with sour crude slates. So bacteria play a leading role in determining the floc structure. And a lot of that is due to the production of EPS. EPS is kind of a glue-like material. It's very important in holding bacteria together in the floc. So it's an important thing. You have to have some amount of EPS. It consists of polysaccharides and proteins, lipids, and nucleic acids. So it's a combination of a lot of different biomaterials. But having excessive EPS, too much EPS, is the most common biological problem globally in refineries that process sour crude. And it's a result of the stress. So we see that all the time in refineries. And unless you have a microscope, it's pretty difficult to understand or to identify it. So a lot of places might go for a long time having EPS issues without really being able to identify the problem. Very simply, having a microscope is an easy way to do that, and I'll show you some slides in a little bit. It can come up in different conditions, different situations, and again, the slime can be visible either with an India ink stain or with a phase contrast microscope, and we'll show you some images around that. EPS is a hot research topic. I just came across this paper yesterday. It is called Global Insights into Extracellular Polymeric Substance from Activated Sludge. This paper was just around municipal wastewater treatment plants. They had somewhere around 15 or 20 samples from around the world and just did some comparative analysis. And I don't mean to get into the nature of this paper in particular, other than to mention that there is a lot of research going on. I'd like to see more of it in industrial wastewater treatment because that's often where it causes a lot of issues. What do you think, Mike? Should we go with the try?
Give it a try. Give it a try. Yeah.
We'll try it. And I appreciate everyone's patience as we try to do this. Let's see. Let me get to the poll. See if we can pull this up.
Yeah. No, it's come up as names not recorded, results shared. So I think it needs to be reset, maybe, because we ran this prior.
Yeah. I'm going to close it and then see if it can reopen. Reopen poll. That's how I did it the last time.
Yeah, it's still coming up with the
No. Okay. All right. Well, we will. The question there, and again, was just around how confident you might be in understanding EPS. If you know about EPS, if you can identify it, if you know the difference between what we're going to talk about next, being tightly bound and loosely bound EPS. So there are two types of EPS, and it's important to know the difference if you have this at your plant. So one is called tightly bound EPS, and that's where the EPS is very close to the cell. It's almost like a capsule or a surrounding the cell. In this picture, it's this part of the microscopic image here where you see this tightly bound EPS. And these would be individual bacterial cells that every cell has this kind of glue or this kind of gel that's around it. We often see that in high food to mass ratio where there's a lot of food coming into the system. Sometimes where there's a nutrient deficiency is when we see this. And sometimes this can be, 10%, 30%, 50% of the biomass. Because of this gel matrix, it's very hard to dewater. It doesn't settle very well. and it can just cause a lot of operational problems when you're trying to manage an activated sludge plant. The second type of EPS is called loosely bound EPS. I'll show you what that looks like. In this microexam image, you can see some down in the lower left, but on the right-hand side is where you see most of it. And this is different. It just looks like a whole matrix of jello or some sort of gel, and then you have bacteria that are spaced out within that gel matrix. This is even harder to dewater. Some people call this slime bulking, and it's caused by stress. And it's a reaction of the bacteria to kind of store up their reserves. This helps them be protected from toxins that are out into the environment or any other environmental changes. And we often see this in systems that have a longer sludge age. So you won't see it in systems less than 10 days, but in a refinery, you're often running longer sludge ages. So it's not uncommon to see that, to see these conditions. And then when you have stress, that's often what happens with biomass. Again, very difficult to dewater, and it doesn't settle very well. So that can lead to increased chemical usage. So if you see your chemical use going up, look under the microscope, see if you're seeing more EPS. It increases the sludge disposal cost because you can't dewater it as well. It can also cause problems with nitrification because nitrifying bacteria like to attach to a dense floc. And if the floc isn't settling very well, you can have a washout of the biomass and a washout of your nitrifying bacteria. And that can be a result of EPS. You'll often see a foam or a lighter scum on top of the DAF or the aeration basin or the clarifiers. Can lead to a suspended solids effluent violation, cost labor to remove that scum that's on the surface, so that costs, it's expensive. And then it can also cause oxygen transfer issues. And when there's less oxygen transfer, that can stress nitrification and BOD and COD removal. Can also cause issues with whole effluent toxicity testing, especially if there's poor nitrification and you get a buildup of ammonia. So sometimes these issues can kind of cascade into one issue into another issue that causes toxicity issue. But those are some of the issues. And again, if you're not used to seeing EPS, you're not looking under a microscope, if you see this, if you see an increase in polymer usage or an increase in or decrease in settling, that could be the cause. It can be, looking for the causative factors, could be loading or feed variability, either from an increase in organic loading or inorganic loading, a change of the crude slate, an unknown chemical dump that could come somewhere upstream without knowing really where it came from. Nutrient and chemical imbalances from low nutrients, either nitrogen or phosphorus, pH swings, or if someone's doing a chemical trial upstream, a new chemical coming into the plant can cause issues. We mentioned before temperature, high or low temperature, or seasonal shifts. During the early summer when things start to get very hot, that's probably more the issue where you're watching from in Asia. As things start to get hot, that can cause issues, especially around stress for an EPS can be a result of that. And then when there's a process shutdown or issues with aeration or mixing or operational changes, or if the DAF isn't working well or APIs aren't working well, equipment problems, those stresses can also lead to EPS. So certainly something to watch out for. Approaches to treating EPS. It can only be removed by wasting. So we can't add a bacteria to degrade it or add something to chew it up or break it up. It can only be removed by wasting. So that's going to take some time. It's going to take some understanding of what the source is, what the issue is in trying to do that. So the first case for a tightly bound EPS, we would increase the wasting to increase wasting it out of the system faster. We would often recommend bioaugmentation to build up a healthy biomass as you're wasting out biomass at the same time. Especially for tightly bound EPS, where a lot of times it's a degradable shock. So bioaugmentation can help with that. And then if you have the capability to introduce step feeding, sometimes distributing the load of BOD throughout the basin instead of having it come in all at one place where you have a very high FM. Sometimes introducing step feeding to distribute that load can reduce the buildup, but I see a lot of plants doing that more and more. And then the same with loosely bound EPS, only can be removed by wasting. And because this is caused by stress, you really want to do some detective work to find out what the causative factors are. So what's causing the stress, and then try to manage that stress. At the same time, you want to increase wasting. So you remove the loosely bound EPS from the system. And at the same time, bioaugmentation can be used to get a healthy biomass in, especially as you're increasing the wasting. Can help improve plant stability, degrade some toxic compounds like phenols and amines, and reduce stress so that this loosely bound EPS doesn't come back. Typically in a refinery, we have a product called BioRemove HC. It's one of many products that we have, but this works well for hydrocarbons and some of the toxic products like phenols and other things that come down into the waste stream in refineries. So this is a good all-around product that I would just highlight for refinery usage. So to summarize EPS, globally, again, this is the #1 biological issue that we see in refineries that are processing sour crude. And again, today, most refineries at some point are processing sour crude. And I guess the poll that we did, we're able to get successfully earlier in the program that confirmed that, at least with this crowd. It can lead to higher chemical costs, discharge violations, and hard to manage if you're unaware of the issues. So again, having a microscope available, pretty easy to train somebody to use the microscope to identify EPS. We work with a lot of partners that do this on a regular basis. Can be very helpful in managing EPS. Again, because it won't come on, sometimes it won't come on all at once. It might come in slowly, and if you're looking at it on the microscope, you can see it coming in and make changes before it's really a big problem. Again, you have tightly bound and loosely bound EPS and different ways of handling those situations. So the next thing we'll talk about, and I think we're doing pretty good on time, is nitrification, some of the challenges and control strategies for nitrification. Okay, the nitrogen cycle in nature. You have natural processes that convert nitrogen into a variety of different forms as it goes through the atmosphere in plants and animals and humans and food and artificial fertilizers. You have nitrogen and nitrite, nitrate, ammonia, all these different forms going through the environment. And these are natural processes, and the wastewater treatment plant takes advantage of those processes, and we'll just skip this poll around nitrification. The question was going to be, how often do you see nitrification issues? And that could be, there was a lot, often, not very often, don't know. So think about that on whether nitrification is an issue that you see at your refinery. We often see it in refining and a lot of different industrial wastewater treatment processes. So just like in the nature cycle of the nitrogen cycle, you have a lot of different forms that nitrogen can take in a wastewater treatment plant and different processes that convert it from one form to another. Ammonification is a process where you have other amines or other organic nitrogen that bacteria can convert into ammonia. You have some processes where bacteria or the biomass will uptake ammonia into the biomass and use that for nutrition and convert that back into organic nitrogen. You have some bacteria, ammonia oxidizing bacteria, that can convert ammonia into nitrite. You have nitrite oxidizing bacteria that convert nitrite into nitrate, NO3. And then you have the denitrification process. So we're talking here about nitrification, but there can also be total nitrogen removal. And that requires the denitrification process. That's where bacteria convert nitrate into nitrogen gas, and that nitrogen gas just goes off into the environment. So these are the processes that we keep in mind when we're looking at total nitrogen removal. When we're looking at nitrification, it's a really sensitive process, but it can be easily troubleshooted. So if you're looking at nitrification, these are really the key parameters, and this is one of the key slides in this whole presentation around nitrification. In wastewater treatment, the ideal range for pH is between 7 and 8. If you get below 6.5 or above 8.5, that's often where you start to see trouble. And that's why I say start to see trouble. Sometimes you can nitrify below 6.5 or above 8.5, but typically that happens over time where you get an acclimated biomass that gets used to those conditions. If it's jumping up and down, that's where you often see issues. Having dissolved oxygen greater than 2.0 milligrams per liter is kind of a good practice to have. If it's below 2.0, sometimes that's not enough, especially in industrial wastewater where you have oxygen, can have oxygen transfer issues. The ideal temperature for nitrifiers is somewhere between 28 and 30 degrees, although plants, you're going to see a wide variety. Start to see issues in the low temperature when things get below 15 degrees. It's where you start to see more issues. And above 35 is where you start to see a drop off on the higher side. Although I've seen plants nitrifying at 40 degrees or even higher than that. Again, if you're getting into those temperatures, hopefully that's happening over a very, very slow period of time, and the bacteria can acclimate to that. Alkalinity is important, and again, something that's sometimes overlooked. Bacteria require carbonate alkalinity, 7.14 kilograms of alkalinity per kilogram of ammonia nitrified. So we typically look for an effluent residual of at least 50 milligrams per liter. If there's no residual or lower than that, that could also be a sign of potential trouble, not only for alkalinity, but for pH control. Phosphorus, we just look for some residual in the effluent. If there's no residual, bacteria require phosphorus for growth, not a lot, but some. And if it's limited, that can cause issues. The BOD and COD needs to be low enough so that the nitrifying bacteria can compete for oxygen. If you have a high BOD, say greater than 50 milligrams per liter, that means biomass is going to be mostly using that oxygen to degrade the BOD. So the BOD really needs to be low enough so that the nitrifiers can compete for that. Sludge age, again, typically in refineries, we're going to be looking at over 10 days. You can nitrify lower, but typically in a refinery, we're going to look at that as being over 10 days. If it's over 30 days, some refineries run with a very long sludge age, 35, 45, 60 days. That can cause other problems where you have toxicity building up in the sludge. So we'll talk about that in a little bit, but that's something to watch out for. And then the last one is just making sure you have a good, healthy flock that settles. You want nitrifying bacteria like to form onto flock. And if they're just out without attached to a flock, they might not settle in the clarifier and they might just wash out. So that's a potential issue. You want to have good, healthy, settling biomass to maintain the nitrifiers. So this is kind of a key checklist. This is really, when we talked about data and looking at data and trying to understand how the plant is operating and to describe problems. If you're seeing a nitrification issue, almost all the time it's going to be one of these issues that's going to be causing the issue for nitrification. So when should you consider bioaugmentation? We have a product called BioRemove AM. It's a very concentrated form of nitrifying bacteria. And when you have one application would be when you have frequent upsets with a slow recovery. So your system is able to recover, but it just takes a long time. Sometimes it can take 15 days, 20 days to recover. If it's taking you that long to recover, I would consider that a slow recovery where you might consider bioaugmentation. Where you have a history of nitrification, but all of a sudden you're unable to recover. That means something changed. You want to find out what that issue is before you add the product. but at least you have a history of nitrification, and you want to be able to make sure your plant's in order before you add the product. And then if your plant has no history of nitrification, you really want to do some investigation work to get it into the condition where the plant's going to be receptive to nitrification. This is typically what we see is that, in an upset recovery, the cause of the upset could be toxicity or a BOD spike, a spike of ammonia, temperature, pH, the same things that we just talked about. And I would say a slow recovery is anything that takes longer than 1 to 2 weeks. I'm going to show an example of some real data for a plant that lost nitrification for about 20 days. This is their effluent ammonia concentration, somewhere around 4 or 5 ppm, and then went up to 40, stayed at 40 to 30 for a couple weeks, and then ultimately they regained nitrification. When we look at that data, we compared it to the nitrification checklist, and there were a couple different things. We looked at DO. Here's the DO, this DO on the right axis, somewhere around 2 to 3. No real changes, especially, when this upset occurred. COD, no increase in loading, so it didn't seem like it was COD. We did see a drop in alkalinity, and we saw a drop in pH. And that seemed to be the issue, is when the pH dropped, that also caused a drop in alkalinity. pH went down to below 6 for a couple days, and that's what it was. That's what caused the die-off of the nitrifiers, everything to come back. They also monitored phenol at this plant, no spikes. Temperature was kind of stable. So we went through that checklist and determined it was pH, and once the pH got back into its normal range, it just took a long time for the nitrifiers to recover. So again, this is larger scale with the pH, again, showing that drop down to about 5.5, and that's what caused the issue at this plant, just that low pH dump. And again, they did not use nitrification, and they did not use bioaugmentation. They had in other occasions, and typically their recovery period is about 7 days or so. So in this case, if they would have added bioaugmentation and recovered as they normally did when they started using bioaugmentation, that would have saved about 2 weeks off of that recovery time. And that's typical, typically what we see, application, typically adding product over the course of a week or so and seeing that recovery come in within 1 to 2 weeks, often saving several weeks when it's a lot slower to come in. So that's an example of nitrification. If the plant is unable to nitrify, but it has in the past, you really want to do some detective work. Is there a new kind of crude? Are there new processes running? Go through the nitrifier checklist, do a micro exam. In this case, this is a sign of stress. When you see something like this, it's called a testate amoeba. This organism here is a sign of very low toxicity. So this would be a good opportunity to show that the system is actually pretty healthy. And then with the microbial community analysis, you can do analysis. You can see, are there ammonia oxidizing bacteria, nitrogen oxidizing bacteria, nitrite oxidizing bacteria. You can see what the different genuses are. And I would recommend not only doing that when you have trouble at the plant, but also do it when things are running well. So you get a good baseline. When you have good activity, what does your system look like when it's running well? And then what does your system look like, if you're having issues, and then you have something to compare it to. The last thing around nitrification I'm going to talk to is just a quick toxicity screen that you can do sometimes to test toxicity. In this case, this plant had a 35-day sludge age, so a long sludge age. They slowly lost nitrification, and then they lost it. It was deteriorating over time, and then they lost it, and they suspected toxicity. So the idea here with this 3-beaker test is to test the influent at full strength, test a blend of the mixed liquor and effluent, and then also test the mixed liquor at 100%. And in each case, we're just going to put a small amount of BioRemove AM so that we know the system has good nitrifiers or know that each beaker has good nitrifiers. And this is the result of this test. This is real data from customer data. The mixed liquor did not nitrify. So it started around 50. We started each of these at the same ammonia concentration, very little nitrification. The mixed liquor and influent didn't get all the way down. The influent fully nitrified overnight. So we measured this for a couple hours in the afternoon and then checked it again the next day. And what this is suggesting is that there's a buildup of mixed liquor, of toxicity in the mixed liquor. So when the mixed liquor was at 100%, it was too much for the nitrifiers. They did not work here. But with the influent, there's nothing in the influent that was really inhibitory. So the idea was to increase the wasting. We had them repeat this test until the mixed liquor was able to nitrify. And that's when we added the BioRemove AM to that plant, and they were able to successfully nitrify. So there's some quick and easy tools that you can do sometimes to screen some of these out. Toxicity sometimes will be harder to screen for. So just to sum up nitrification, it's probably the most common bioaugmentation application that we see in refineries. There's a nitrifier checklist. It's a good way to review, check data. Microexam can help to look for stress indicators. DNA analysis can show what organisms are present or absent if there's an issue. And toxicity is relatively simple to screen for. BioRemove AM can be used together with BioRemove HC for toxicity reduction and enhanced stability. And don't add nitrifiers if the system isn't ready to nitrify. So if you go through the checklist and there are some things that you just can't rectify, you can't change the pH, you can't get the DO to be above 1, don't add nitrifiers because the system it's not ready yet. So make sure that checklist is ready to go. If you want to try bioaugmentation, make sure you can get through that whole checklist and make sure the system's ready. Given the time right now, we have 10 minutes left. I'm going to go through a couple slides very quickly, and then we'll get into some questions. So settability issues, again, this could be a whole webinar on its own with filamentous bulking. Again, using a microscope to identify filamentous bulking, to identify the different filaments, can then help you identify the causative factors for those different filaments. There's a lot of different control strategies that you can use if you know you have a low FM, low dissolved oxygen, septicity or organic acids, or nutrient deficiency. Filaments can help point you in that direction to say, are these some of the common issues? Often in refining, we see pin flock, very small, weak flock, and that leads to poor settling. A lot of that is due to old sludge age. A lot of refineries, again, run a longer sludge age. If it gets too long, sometimes inhibitory compounds can build up. Sometimes there can be shearing from high turbulence that break up the flock particles. So some of the considerations, consider the sludge age. Bioaugmentation can help promote better flock formation and add coagulants to improve flocculation if that's an issue. So again, other issues that can happen in refining, that could be a webinar on their own, different organic and hydrocarbon loadings, different biodegradation and loading characteristics, inorganic stressors, including hydrogen sulfide, and biological indicators like toxicity and oxygen deficiency. The last thing I'll do is just run through a couple of the products that we have that can be used in refining. We talked about BioRemove AM and BioRemove HC. BioQuick 5140 is a good supplement for starting up wastewater treatment activities or recovering from an upset. OdorCap 5700 controls sulfides and VFA-based odors. We have a variety of nutrient blends and some other products that go well in refining. Again, we have a global team. Mike McWilliam is in Japan, Director of Sales. We also have Vivek in India, if anybody's calling in from that region. And then we'll finish up with questions. So we have a couple minutes. And Mike, can you go through the questions and see if any questions have come in?
Yeah, and feel free to add any additional questions to the list using the Q&A tab at the top for everyone online. And those that we don't get to today, we'll make sure we'll follow up with you afterwards as well to provide additional feedback. So, Matt, thank you for going through that. It was really valuable and sharing that experience. So some questions we've got here. One was, what type of microscope is needed to do microscopy work to check, say, the biomass and the EPS?
Yeah. So one of the key factors for choosing a microscope is making sure that it's a phase contrast microscope. And a phase contrast does something with the light that makes it easier to differentiate the flock against the background. A lot of times we'll see what's called a bright field microscope, where it's just a light at the bottom. And you can see bacteria, you can see some filaments, but a lot of things are difficult to see with a bright field. And the examples that we had here in this seminar here, this is with a phase contrast microscope. You see this blue background, and you see the flock looks almost brown or tan, and you see these dark blue filaments. You generally don't have that same sort of differentiation. So phase contrast. And then from the magnification level, you want to look at a couple different levels. One at the 100x, so that's a 10x ocular lens, and a 10x lens on the microscope to make 100x to look at the flock. So that gives you a broad range over the whole biomass. That's where you can see flock particles, you can see the higher life forms, you can see in the bulk water. Most microscopes then have a 40x and then also a 100x lens that makes it 400 and 1,000 total magnification. And that's really needed to identify the different filaments. So when you get into filament identification, it's really looking at the morphology, the size of the filaments, the shapes of the cells, whether they grow end to end, whether they're round or rectangular shaped, the size of the filaments. So all those you can only tell using a phase contrast microscope at 1,000 magnification. So phase contrast and up to 1,000. They're a little more expensive, but certainly worth the investment.
Thanks, Matt. That's good. You talked about nitrification. Can you share a bit more around what wastewater temperature will start to impact nitrification? Because in Asia, often, the temperatures of wastewater systems can be much higher than other regions.
Yeah. So the rule of thumb is kind of around 35 is where things start to deteriorate. 30 is the optimum. And there are growth curves that you can see that show 30 is the optimum and then show a drop off. But it doesn't drop off immediately. And again, as I said, especially if the change is slowly over time. I've seen some refineries nitrifying up to 40 degrees. But if the change happens quickly, like over the course of a week or two, as higher temperatures come in, or if there's a change in the refining process, or something happens upstream where you get a higher temperature that come in, that can happen pretty quick. So it's the absolute temperature is part of the issue, but also the degree to which it changes. So keep an eye on things once they hit 35. That's where they can start to fall apart. But it can still continue up to 38, 40, 41, 42 even.
Perfect. Thank you. Thank you for sharing that. Can you share any examples of specialized treatment of refinery wastewater systems, application specific?
Yeah, so what I talked about here mostly is, typical issues that come on a day-to-day basis of just, influent coming in, effluent going out, and you're treating that mixed stream that comes in. A lot of times at refineries, they might have a highly toxic, high-strength stream that's kind of kept on a side tank that's often very expensive to dispose of. We've been involved with a lot of applications treating high-strength waste or kind of preparing the biomass to receive low volumes of high-strength material. So working with the wastewater treatment plant to get it in a situation where it can receive amounts of higher strength material, whether that's high nitrogen stream, high organics, high toxicity, and then trying to bleed that through the wastewater treatment plant rather than have an expensive way to dispose of it. So that's some of the issues. And again, those are really case by case. So I would reach out to either your distributor or Mike to help you assess, whether or not that's an opportunity for you. Not doesn't work all the time. It's not for every case, but if there are some things we can do treatability study wise beforehand to improve the chances, we can help review some of that work.
Excellent. Appreciate that. So we're at the top of the hour. I wanted to pass on again our appreciation for everyone joining today. And we hope that this was useful, and please do not hesitate to follow up. You can reach out through the email or through the EnviroZyme system. There's lots of case studies and information about the products on our website. And we look forward to having you join our next webinar in the near future. Thanks again, Matt, and everyone for joining.
And thank you. And feel free, I've got my LinkedIn connection here. If anybody's interested in that, I'd love to connect with you. Thanks again. Have a great afternoon and good evening.
Good afternoon, everyone. Bye.